Hi everyone, and welcome to my presentation about ecotoxicological studies of bioplastics with daphnids and algae. My name is Silja Denise Kröger and I'm a PhD student here at Hamburg University of Applied Science. My research fields are the pollutant release of organic pollutants from bioplastic materials and conventional plastic materials, and in addition also ecotoxicological studies. So here I give you a short overview about the tested materials. We tested 4 different materials, and in the first column of the table you can see the name of the materials, and the first abbreviation of the name always for every material is BPE, which stands for the project. And then we have got the application for the material, so MG means Marinegeo material, FC stands for fishing crates, SP for soft packing, and FB for fishing bait. And the last abbreviation is the polymer from which the material is made of, so PLA is polylactic acid, PBS is poly 1.4 butylene succinate, and PHBV is poly 3-hydroxybutyrate co-3-hydroxywalerate. And under the table you can see the chemical structures of the monomers. So first I would like to start with the materials and methods we used for the daphnid testing. So Daphnia magna as test organism was used, and here on the right side you can see a daphnid under the light microscope with a magnification of 200. And we performed 2 different types of tests. We performed acute toxicity tests according to OECD guideline 202. And these tests last 48 hours, and the immobilization of the daphnids was observed. In addition to acute testing, we also tested the bioplastic materials by chronic tests according to OECD 211, and the test duration was 21 days for chronic testing, and the reproduction of the daphnids was observed. The plastic concentration was between 0 and 10 gram plastic per 1 liter medium. This is in accordance with the study of Lithner in 2009. So here you can see the experimental setup for the chronic daphnid and acute daphnid test. And we have thin plastic films from the bioplastic materials, which were about 100 micrometer thick. And first of all, we cut them into small pieces of 1 to 1 centimeter. One part of these films were tested directly in contact to the daphnids, as you can see on the left side. We put the plastic sheets, the small plastic sheets, directly into the test in clean beakers and added some media, and then we put the daphnids to the test. And we also tested the leaching of pollutants from the bioplastic materials, and therefore we put the thin plastic sheets into a bottle with medium, and they were shaken for 14 days in the dark, and afterwards only the medium without the plastic sheet was tested in the acute and chronic tests. After the tests ended, we also evaluated the size and the color of the daphnids, because if they are very small or the color is very light, this may be a reason, or this is an indicator for bad environmental conditions towards the daphnids, and therefore we observed every daphnid by light microscopy with a magnification of 200. The daphnids were removed from the media, and so only the media was extracted by liquid extraction with hexane. This is made because we wanted to analyze the chemical pollutants which were leached out of the plastic materials. And after the extraction, the extracts were analyzed chemical by GCMS, gas chromatography, mass spectroscopy, with regard to organic pollutants like phthalates or bisphenol A. So now I would like to explain you the results from the daphnid tests. In all acute daphnid tests, we didn't observe any immobilization of the daphnids. So no daphnids died during the 48-hour testing, and also no deviations from the negative control in size and color was observed. So compared to other studies, like for example the study of Lithner et al. From 2012, the bioplastic materials provoke less effects towards daphnids than conventional plastics. So in this table you can see the results for the chronic daphnid test with a duration of 21 days. And as you can see, during the chronic tests, a significant poor offspring for all materials in contact tests was observed. But we didn't observe deviations in size and color, and also we didn't find any pollutants in the media. So, but as you can see in the table, the leaching test, the chronic leaching tests provoked stronger effects towards the daphnids than the chronic contact testing. And this could be explained by the sample preparation, because during the shaking process of the plastic material in the medium, the pollutants from the material migrate to the water phase, and so they become more bioavailable for the daphnids. And this is in accordance with a study from Schrank in 2009. So there the reproduction of Daphnia magna was also influenced by toxic leachates, not by the plastic itself, but by the leaching compounds of it. So now I would like to explain a little bit more about the Marinegeo material PLA. And as you can see, in leaching tests, in chronic leaching tests, we didn't have any offspring, and also all adult daphnids died between day 7 and day 17. So this was a really strong reaction of the daphnids towards the material. And we analyzed the media by GCMS. And then we found out that 1 liter medium contained 1 gram plastic, contained 186 microgram methylnaphthalene. And methylnaphthalene is known as a toxic substance for aquatic organisms and humans. So we didn't have an explanation for the presence of methylnaphthalene in the samples. So we were in close contact to the producers, and they sent us 2 more samples of the material. And in these samples, we didn't find methylnaphthalene. So the question was why the first sample of the material was contaminated with methylnaphthalene. And this could be explained by the production of the films, because therefore the material, which was delivered in pellets, was heated and rolled into thin films, and for the rolling machine lubricating oil was used, and these oils often contain organic pollutants like methylnaphthalene. So here are the results for the chronic tests of the PBS material, and as you can see, we had a high amount of offspring with 27 neonates during the test period. And also we observed a significant higher growth rate of the daphnids during the test. So the adult daphnids growth stronger than the daphnids from the negative control. And this indicated endocrine effects. So we also analyzed the medium, and we found 215 micrograms succinic acid in 1 liter medium. And succinic acid is a compound which is also produced in the Krebs citric acid cycle of cells. And from succinic acid, a kind of energy was produced. So this might be the reason why we have such strong growth effects of the material. Yeah, and in addition to the daphnid tests, we also carried out tests with the marine algae Phaeodactylum trichinotum. This is a microalgae and belonging to the group of diatoms. And on the right, you can see the algae under the light microscope with a magnitude of 400. These tests were carried out according to ISO 10253 from 2006. This is nearly the same like OECD guideline 201. And the test lasts 72 hours. And during the test, the reproduction of the algae was observed every 24 hours. And this is made by the evaluation of the fluorescence of the algae. So on this sheet, you can see the experimental setup for the marine algae test with Phaeodactylum trichinotum. In contrast to daphnid tests, we only carried out leaching tests with the algae because for contact testing, it is necessary to introduce the plastic material directly into the algae culture, and we are observing the fluorescence of the algae. So if there are plastic sheets or particles in the culture, the fluorescence is weakened by them. So we only tested leachates of the bioplastic materials with the algae. We tested concentration of a maximum of 50 gram bioplastic per 1 liter artificial seawater. And like for the leachates for the daphnids, the plastic materials were cut into small pieces and then put in the media and shaken for 14 days in the dark to avoid disintegration of the materials. Afterwards, the leachates were diluted by a two-step procedure until the lowest concentration of 1 to 128 was reached. And this is a concentration of 0.4 gram bioplastic per 1 liter medium. To avoid influences by external effects, like for example uneven light conditions during the test, the test concentrations or the leaching dilutants were distributed randomly on the well plates, which you can see there. Afterwards, the algae were added to the plate. And for every concentration, one well was only filled with the media and without algae, because for the extraction of the media. So these wells were extracted by liquid-liquid extraction with annexane and afterwards analyzed by GCMS. And the plates were analyzed with a Tecan reader. So the fluorescence was observed every 24 hours. And at the end of the test, a dose-response curve for all materials was prepared, and I will explain them on the next slide. So on this slide, you can see the results for the PLA material, which should be used as Marinegeo material. And on the right, you can see the dose response curve for that material. So on the x-axis, there is the concentration of the plastic in gram per liter, and the axis is in a logarithmic scale. And on the y-axis, you can see the growth inhibition of the algae. And during the 3 replicates, so in all 3 tests, no growth inhibition of the algae was observed, and also no deviation of the algae from the negative control in cell shape and color was observed. And we also didn't find any pollutants by GCMS analysis. So here are the results for the PLA material for the fishing crates, and like for the Marinegeo material, we didn't observe growth inhibition and also no deviations in cell shape and color from the negative control was observed. So in addition, we found no pollutants by GCMS analysis. And so what we can say about the PLA materials. The PLA materials seems to be not toxic to marine algae. So here are the results from the marine algae test for the PBS-based material, which should be used as soft packing. And like for both PLA materials, we didn't observe growth inhibition and also deviations from the negative control. But we found about 200 micrograms succinic acid per 1 liter medium by GCMS analysis. So succinic acid might be a degradation product of PBS. But in this case, it seems to be not toxic to marine algae. So, and here are the results for the fishing bait material, so for the PHBV material. And in contrast to all other materials, we observed growth inhibition for the algae. So as you can see in the dose response curve, for all 3 replicate, a growth inhibition was observed. And so we were able to calculate a EC50 value, which means the effect concentration where 50% of the organisms showed an effect. And the EC50 for PHBV material was 6.3 gram plastic per 1 liter medium. By light microscopy, we didn't observe deviations from the negative control in cell shape and color. And we also didn't find organic pollutants by GCMS. So it might be that there are other anorganic pollutants, but we only observed, or we're only looking for organic pollutants. So here's my conclusion. For bioplastic and conventional plastic materials are less toxic data available. This is because every producer have got an individual composition of the plastic material, and sometimes there are more than 1,000 different chemicals in one plastic material. So in this study, different organisms show different effects toward the plastic material. So it is necessary in ecotoxicological studies of plastic materials to use different species of test organisms. And in this study, we combined bioassays and with chemical analysis by GCMS, and this combination contributed to the result interpretation. Like, for example, we found out that we had methyl naphthalene in one sample. But the good thing also of this project is that we are in a really close contact with the producers. So we found out that we have a pollution of the material, and then we were in contact, and now the material was tested again. And for the PHBV material, for the fishing bait material, the material will be improved by the producer so that it will be not toxic for the marine algae. And what we can say finally, compared to other studies, bioplastic seems to be less, or this kind of bioplastics used in this project seems to be less toxic than conventional plastics. So yeah, thank you for your attention, and please don't hesitate to ask any questions.